Each cell has its own chromosomes, and these chromosomes are composed of many gene codes. How does the cell convert these gene codes into an amino acid, which is the building block for protein? Through the process of translation. Hi guys, this is Angela from 2Bio6, and today we're gonna talk about the process of translation. Translation occurs right after the DNA is converted into mRNA through the use of the RNA polymerase through a process called transcription. So what is translation? In translation, the ribosome translates the sequence of messenger RNA molecule to a sequence of amino acids during the synthesis of proteins. So now, let's have a closer look at the process of translation. Just like any movies, which have a beginning, middle, and end, the process of translation also has three parts, namely initiation, elongation, and termination. Are you ready? Let's begin. So first is initiation, the phase where the ribosome gets together with the mRNA and the first tRNA so translation can begin. But first, we need a few ingredients. A large and small ribosome, a mRNA with instructions for the protein synthesis, and an initiator tRNA which carries the first amino acid in the protein and is almost always methionine. Just like a puzzle piece, these ingredients should come just in the right way. Together, they are called the initiation complex. It is the key molecule in making a new protein. Now, let's jump into the process. The tRNA carrying methionine attaches itself to the small ribosomal subunit. Together, they connect to the 5' end of the mRNA by recognizing the 5' GTP cap, which is added during processing in the nucleus. Afterwards, they walk along the mRNA in the 3' direction, stopping as they reach the start codon, often the first AUG. However, in bacteria, the situation is a little different. The small ribosomal subunit doesn't start at the 5' end of the mRNA and travel toward the 3' end. Instead, it attaches directly to certain sequences called the Shine-Dallarno sequence in the mRNA. These sequences come just before start codons and point them out to the ribosomal subunit. So why should we use the Shine-Dallarno sequences? These bacterial genes, which are transcribed in groups and are called operons, can contain the coding sequences for several genes. The Shine-Dallarno sequence of a gene is the start of each coding sequence, letting the ribosome find the right start codon for each gene. Next is elongation. In this phase, as the name implies, is when the polypeptide chain gets longer. The question is, how does the chain actually grow? Let's find out. But to do that, we need to take a look at the first stage of elongation after the formation of initiation complex before any amino acid links have been formed. So first, methionine-carrying tRNA initiates out in the middle slot of the ribosome, also known as the P site. Second, a fresh codon is exposed in another slot called the A site. The A site will be the landing site for the following tRNA, one whose anticodon is a perfect or complementary match for the exposed codon. Once the matching tRNA has landed in the A site, the mechanism will begin. Initially, the formation of the peptide bond that connects one amino acid to another. This step transfers the methionine from the first tRNA onto the amino acid of the second tRNA in the A site. So now, we already have two amino acids which can be considered as polypeptide. The methionine forms the N-terminus of the polypeptide and the other amino acid is the C-terminus. But we may want a longer chain. So how is it going to be formed? Once the peptide bond is formed, the mRNA is pulled out onward through the ribosome by exactly one codon. This shift allows the first empty tRNA to drift out via the E site or the exit site. It also exposes a new codon in the A site so the whole cycle can repeat. Last is termination. Sadly, even good movies have to end. Polypeptides like a good movie must eventually come to an end. As you can guess, in this phase is where translation ends. It is when a stop codon in the mRNA, which are UAA, UAG, or UGA, enters the A site. These stop codons are conceived by proteins which are called release factors. These proteins nearly fit into the P site, although they aren't the tRNAs. These proteins mess with the enzyme that normally form peptide bonds, making the enzyme add a water molecule to the last amino acid of the chain. The process separates the chain from the tRNA and thus releasing the newly formed protein. So, now what? Luckily, translation equipment is very reusable. After the small and large ribosomal subunits separate from the mRNA and from each other, each element can, and usually quickly does, take a part in another round of translation.